Welcome to this video on the nervous system histology slides, presented by Visual MD. We begin with the neuron. You should expect to see a large cell body, a prominent nucleus with a clear nucleolus, and basophilic granules known as Nissl bodies. Look for a big, star-shaped cell with a clear nucleus to identify it immediately. Neurons are typically found in the anterior horn of the spinal cord, cerebral cortex, and brainstem nuclei. Their function is to receive, process, and transmit impulses. Motor neurons specifically send signals to muscles. Next are neuroglia, or support cells. They are much smaller than neurons, with dark, dense nuclei and cytoplasm that isn't clearly visible. They are numerous around neurons. If you see many tiny dots surrounding a neuron, those are glial cells. They are located throughout the central nervous system. Neuroglia are divided into cells of the CNS and PNS. In the CNS, we have astrocytes, maintain blood-brain barrier, oligodendrocytes that form myelin sheath, microglia as immune cells, and ependymal cells that produce and circulate CSF. In the PNS, Schwann cells form myelin sheath, and satellite cells provide support and nutrient exchange. Moving on to the cerebellum, look for three distinct layers. The outer pale molecular layer, a single row of large cells called the Purkinje layer, and the inner, very dark granular layer. If you see a line of big flask-shaped cells, you are looking at the cerebellum. This cerebellum's location is in the cerebellum. Its primary functions are the coordination of movement and maintaining balance and posture. Next is the cerebrum, or cerebral cortex. Expect to see an outer gray matter and inner white matter. Neurons are arranged in 6 layers, with prominent pyramidal cells. If layers exist but there is no Purkinje cell row, you are viewing the cerebrum. The cerebrum is located in the cerebral cortex. Its functions include thinking, memory, voluntary movement, and sensory perception. Remember not to confuse it with the cerebellum. The cerebrum has less organized layering. The spinal cord is next. You will see a central butterfly or H-shaped gray matter, surrounded by white matter, and a central canal in the middle. This distinct shape makes it one of the easiest slides to identify. The spinal cord's function is centered on reflexes and the conduction of impulses. An exam tip: the anterior horn is larger, motor neurons, while the posterior horn is thinner, sensory neurons. The lateral horn contains autonomic neurons. It is present in the T1 to L2 segments for sympathetic outflow and the S2 to S4 segments for parasympathetic outflow. Now, the peripheral nerve. It consists of bundles or fascicles of nerve fibers encased in connective tissue layers. The layers are the endoneurium surrounding axons, the perineurium surrounding fascicles, and the epineurium covering the outer surface of the nerve. Identification clues for the peripheral nerve include wavy fibers, fascicles, and connective tissue covering. It is located outside the CNS and carries impulses to and from the CNS. A key exam tip is that it looks like cable bundles and lacks neuron cell bodies. Finally, the dorsal root ganglion. Look for large, round neurons with a central nucleus, surrounded by satellite cells forming a ring. These clusters of large neuron cell bodies often look like, quote, fried eggs with halos. The dorsal root ganglion is located in the dorsal root of the spinal nerve, and its function is sensory transmission. For identification, note that neurons are grouped, unlike the scattered neurons in the CNS, and the presence of satellite cells is a key identifier. And that's it. Nervous system histology made simple. If you can recognize these slides, you're already ahead. Take a moment to like this video, subscribe, and turn on notifications. It helps the channel grow and allows me to create more clear, exam-focused videos like this for you. Thank you for watching, and best wishes in your studies. Stay consistent. Success is coming.